The cell membrane is very flexible and it adjusts its shape to fit its environment. The molecules that make up the cell membrane move in groups and swirl around. The soap film was very flexible, and when we moved it up and down, the film was also going up and down without popping. When you look at the soap, you can see the surface has molecules moving in clumps. The thin film that spans across the cell membrane represents the cell membrane. The finger covered in bubble solution was pushed into the film, and it did not pop. Instead, the film made way for the finger, going around it, and then closed once the finger was removed. This represents the concept that, like the bubble layer, cell membranes can instantaneously repair small tears in the cell membrane. We use the straw to blow a bubble about 6 inches wide into the bubble solution, and then boost smaller bubbles into it. Each smaller bubble creates a compartment of air that is within the large bubble. The big bubble represents the cell membrane, and the smaller bubbles represent the membrane-bound organelles found in eukaryotic cells, such as the mitochondria. The components that make up both these membranes are phospholipids, which create a phospholipid bilayer. Membrane proteins can also drift across the lipid bilayer. These proteins can move rotationally, laterally, and transversely. A phospholipid will rotate in order to interact with its immediate neighbors. It moves laterally to provide a membrane with fluid structure, and it moves transversely to allow asymmetry to be maintained, which is critical for membrane function. The soap bubble modeled this cell concept because it represents how the loop of thread, which is the membrane protein, is able to freely move around the soap membrane after the inside is popped. As the straw leaves the bubble, there's a small gap, which represents the junctions that form between neighboring animal cells. When a gap is open, it represents when the nutrients and materials from different cells that have bonded to the cytoplasm of the initial cell can quickly transfer. Due to the membrane's ability to self-repair, this gap does not last long. The bubble represents the cell, and the string action demonstrates binary fission, cutting the bubble in two. The single cell undergoing binary fission creates two new, genetically identical cells from cleaving due to a thin ring of proteins located at the cell's midpoint, which contract.